Hi everybody, and welcome to our module on the exocrine pancreas. The pancreas is really two organs in one. Part of the pancreas is an endocrine organ. This secretes the hormones insulin and glucagon for the control of blood sugar levels. Another portion of the pancreas is an exocrine organ. So exocrine organs are things that secrete substances into ducts, things like salivary glands and sweat glands, and the exocrine portion of the pancreas is also an exocrine organ. And the secretions that come from the exocrine portion of the pancreas aid in digestion, so it secretes things like fluid, which has many important substances in it, including bicarb to raise the pH in the duodenum, and it also contains digestive enzymes. The exocrine portion of the pancreas contains two cell types, the acinar cells and the ductal cells. Acinus is a Latin word that means berry, so the acinar cells are the cells found in this berry-like portion of the exocrine pancreas, and then the ductal cells are the cells that line the ducts of the pancreas. The acinar cells secrete enzymes, and they also secrete some fluid, although mostly they secrete electrolytes like sodium and chloride. We'll talk more about that in a minute. The ductal cells modify the fluid that is secreted by the acinus as it moves out towards the intestines. They also secrete bicarb, and importantly, the ductal cells are permeable to water, so they add water to the pancreatic juice. One of the most important components of pancreatic fluid is bicarbonate. This neutralizes acidic fluid coming from the stomach. Pancreatic juice is secreted into the duodenum, and when it reaches the duodenum, it will meet with acidified fluid and chyme that is coming from the stomach. So this bicarb helps to raise the pH. And that's very important because, as we will see, the pancreatic enzymes don't work very well at a low pH. So this bicarbonate raises the pH in the duodenum so that the pancreatic enzymes can work. There are also electrolytes found in the pancreatic fluid like sodium and chloride and potassium. The composition of pancreatic fluid varies with the flow rate. So when you are in the fasting state and you haven't eaten any food in a while, there's relatively low flow coming through the pancreatic ducts. And this fluid has a high concentration of chloride and a low concentration of bicarb. On the other hand, when you eat a meal and the pancreas is stimulated, the flow increases. And when you have high flow through the pancreatic ducts, that fluid will have low chloride and high bicarb. It's basically the opposite of the low flow state. Shown on this slide is a fairly classic depiction of how the concentration of different elements of the pancreatic fluid changes with flow rate. On the x-axis, I've shown the flow rate, and on the y-axis is the concentration of different electrolytes in the pancreatic fluid. And all of the electrolytes in the pancreatic fluid start out at the level that you find in the plasma. So this is the sodium level of plasma, for example. This is the chloride level of plasma. This is the bicarb level, and this is the potassium level. No matter how high the flow rate gets, the concentration of potassium and sodium don't change. They're always equal to what you have in plasma. However, the bicarb and chloride concentrations do change with flow rate. When the flow rate is very low, the concentration of chloride is quite high and the concentration of bicarb is relatively low. This is just like it is in the plasma. On the other hand, when the flow rate rises, as we go out on this portion of the x-axis, the concentration of bicarb gets much higher so that it's greater than the plasma bicarb concentration, and the concentration of chloride falls so that it becomes lower than the concentration in plasma. It's important to remember that pancreatic fluid is always isotonic. That's because the sodium concentration doesn't change, and the sodium concentration is what determines whether something is hypo or hypertonic. And I've written that down here to remind you the sodium and potassium concentrations are always the same as in plasma, no matter what the flow rate. The reason the concentration of bicarbonate and chloride change with flow rate is because the pancreatic fluid is modified as it flows from small to large ducts. So as pancreatic fluid moves through the pancreas, it travels first through smaller ducts and then it collects into larger ducts, and I've shown that on this slide here. Bicarb is initially secreted into the small ducts, like I've shown with this pump here, putting bicarb into the smaller ducts. It is then reabsorbed in the larger ducts in exchange for chloride. So bicarb gets reabsorbed in the larger ducts and chloride gets pumped into the larger ducts. What this means is at very slow flow rates, almost all the bicarb will get reabsorbed, and there will be relatively little bicarb in the pancreatic fluid. There will also be relatively high concentration of chloride in the pancreatic fluid at low flow rates. On the other hand, you can imagine when the fluid is moving through here very quickly, there isn't enough time for this pump to reabsorb bicarb. Therefore, the bicarb passes out into the intestine, and the concentration of pancreatic fluid will have a much higher bicarb at high flow rates. It will also have a much lower chloride concentration at high flow rates because there won't be enough time to secrete much chloride into the fluid. And I've summarized that here in the bottom bullet here. Reabsorption is ineffective at higher flow rates. The hormone secretin is the main stimulus for bicarbonate secretion by the pancreas. I talk about secretin in more detail in the module on GI hormones, but for our purposes here, I'll just remind you that secretin is released in response to H+ atoms in the duodenum. Essentially, when the pH in the duodenum gets low, as acid from the stomach flows in, that stimulates the release of secretin, which in turn stimulates bicarb secretion to raise the pH back to the level where the pancreatic enzymes like to function. Secretin is secreted by S cells in the duodenum. Up until this point, we've been talking about bicarb, which is secreted by the ductal cells, shown in red in the drawing on the screen. Now we're going to talk about pancreatic enzymes. Those are secreted by the acinar cells, shown in green in the drawing on the screen. And one of the major hormonal stimuli for release of pancreatic enzymes is the hormone cholecystokinin, and I talk about this in more detail in the video on GI hormones. Another stimulus for the release of pancreatic enzymes is acetylcholine via what's known as vagovagal reflexes. These are local reflex circuits within the GI tract in which both the afferent and efferent fibers are found within the vagus nerve. That's why it's called a vagovagal reflex. What happens is hydrogen ions or amino acids or fats that find their way into the duodenum can activate these vagovagal reflexes, which will send nerve signals to the pancreas to stimulate the release of pancreatic enzymes. Pancreatic enzymes can digest all the different types of foods that we consume in our meals. They can digest carbohydrates and fats and proteins. And there are many different pancreatic enzymes, but I've listed the most important ones on this slide. Alpha amylase is important for the digestion of carbohydrates and starch. We'll talk about that in a minute. Pancreatic lipase and phospholipase A and colipase are all important for digesting fats. And then proteases and trypsinogen are important for digesting proteins. Alpha amylase is a pancreatic enzyme that digests starch. Starches are polysaccharides. They are made up of repeating glucose molecules linked together into long polymers. Specifically, they are made up of repeating alpha-D-glucose molecules built into long chains. Starches are found in plants. Humans do not have starch. Humans instead have glycogen, which is a similar but slightly different structure, which I talk about in the biochemistry modules. And starches are made up of two different polymers of glucose. One is called amylose and one is called amylopectin. This is the structure of amylose at the bottom of the screen here. It's made up of straight chains of linked glucose molecules. Amylopectin, on the other hand, has branching chains of glucose molecules. Now what alpha amylase does is it hydrolyzes or breaks alpha-1,4 linkages. This is a bond between the 4 carbon of one glucose molecule and the 1 carbon of another glucose molecule. So alpha amylase will break this bond here, and that helps to break down the starch into glucose molecules. Amylase is secreted in an active form by the pancreas, and this is somewhat unusual. As we will see, many pancreatic enzymes are secreted in an inactive form, and they must be activated to do their job in the intestines. That is not the case for alpha amylase. There is a salivary or lingual amylase that is very similar to pancreatic amylase. However, its optimal pH is greater than 6, so it works in your mouth, but it is inactivated in the low pH environment of the stomach. Pancreatic amylase, on the other hand, functions in the small intestine, and as we will see, this enzyme is a useful marker in acute pancreatitis. Levels become elevated in the serum, and one of the ways you can diagnose acute pancreatitis is to find an increased level of alpha amylase. When alpha amylase breaks down amylopectin and amylose, it breaks it down into smaller sugar polymers like maltose, which is 2 sugars in length, or maltotriose, which is 3 sugars in length, or structures called limit dextrans, which are short polysaccharides that have a branch in them. Glycogen can also be broken down into limit dextrans, and we talk about finding these in certain glycogen storage diseases. In the case of starches that we consume in our diet, limit dextrans are just one of the breakdown products of the amylose and amylopectin. Now, as you may know, maltose and maltotriose and limit dextrans cannot be absorbed by the intestine. Only monosaccharides are absorbed by the intestine. So all carbohydrates must be broken down into glucose, fructose, or galactose in order to be absorbed. So once alpha amylase breaks down amylose and amylopectin, the digestion of carbohydrates is not complete. Further digestion has to continue, and this occurs at the intestinal brush border. There are enzymes called allosaccharide hydrolases like maltase and sucrase and lactase, and these break down those short polymers of glucose molecules into structures like glucose and fructose and galactose, which can be absorbed. And it turns out that digestion at the brush border is the rate-limiting step for carbohydrate digestion. So alpha amylase can quickly break down those starches, but then they have to wait to slowly work their way through digestion at the brush border in order to be absorbed as one of these structures right here. And as you may know, glucose, fructose, and galactose all have the chemical structure C6H12O6. They are just built differently from one another. They are all isomers of glucose. Now let's talk about pancreatic fat digestion, and the most important enzyme here is pancreatic lipase. This hydrolyzes the 1 and 3 bonds of triglycerides, so let's talk about what that means. This is the structure of a triglyceride molecule here. It's made up of glycerol, which is a 3-carbon structure, and then 3 long-chain fatty acids. What pancreatic lipase does is it lyses off 2 fatty acid molecules and leaves behind 1 fatty acid attached to glycerol, and this is called a monoglyceride. So fatty acids and monoglycerides are the breakdown products of pancreatic lipase. This enzyme, just like amylase, becomes elevated in the serum in acute pancreatitis, and we often use this as a marker of acute pancreatitis and to make the diagnosis of acute pancreatitis. Another pancreatic enzyme for fat digestion is called colipase. This assists pancreatic lipase in doing its job. And then finally, there's a pancreatic enzyme called phospholipase A2, which is important for hydrolyzing phospholipids that we consume. It is secreted as an inactive enzyme called prophospholipase A2, and this is activated by trypsin. And as we will see, many of the enzymes that digest protein are also activated by trypsin. It is an important activator of a number of pancreatic enzymes. So now let's talk about the protein digestion enzymes. There are a number of these. These include trypsin, chymotrypsin, elastase, and carboxypeptidases. And all of the protein digestion enzymes are secreted as proenzymes that are inactive. These are called zymogens. The way the protein enzymes are activated is through the action of trypsinogen. As I told you on the last slide, trypsin is one of the pancreatic protein enzymes, and it is secreted as an inactive zymogen called trypsinogen. Trypsinogen can be activated to trypsin by a brush border enzyme called enterokinase. I've shown this down below. Enterokinase can convert trypsinogen into trypsin. Trypsin is then a very powerful pancreatic enzyme because it can activate all the other protein enzymes. For example, it can convert chymotrypsinogen into chymotrypsin. It can also convert proelastase into elastase, and it can also convert procarboxypeptidase into carboxypeptidase. It can even go back and activate more trypsinogen to become trypsin. So it is thus a very powerful protein enzyme. It can activate all the other protein enzymes, and as I told you before, it can also activate phospholipase A2. There's a very important clinical tie-in with the pancreatic physiology we've been discussing, and that has to do with acute pancreatitis. Acute pancreatitis is inflammation of the pancreas. Patients with this condition develop epigastric pain, nausea, and vomiting. And I talk about the clinical syndrome of acute pancreatitis in detail in another module. But for our purposes here, I want to remind you of the underlying pathophysiology of acute pancreatitis. In this disorder, there is blocked secretion of enzymes while synthesis of pancreatic enzymes is ongoing. What happens next is that large amounts of trypsin appear to become activated, and as we have shown before, trypsin can activate more trypsin. It can also go on to activate phospholipase and chymotrypsin and elastase. All of these enzymes are capable of digesting proteins and fats, and they will begin to digest the proteins and fats inside the pancreas itself. This process is called autodigestion. Essentially, the pancreas begins consuming itself. And this is one of the reasons that acute pancreatitis is such a severe disorder. Once it gets going, the enzymes continue the process by digesting more and more of the pancreas. It's very high yield to remember for step 1 of your boards that autoactivation of trypsin is important in the pathophysiology of acute pancreatitis, and that there is autodigestion by the pancreatic enzymes that occurs. Another tie-in with acute pancreatitis has to do with diagnosis. We diagnose acute pancreatitis by identifying elevated serum levels of pancreatic enzymes. Specifically, we look for increased levels of pancreatic amylase and lipase. Both are elevated in acute pancreatitis. They can both also be elevated in other conditions. Lipase is more specific for pancreatic damage, but we will check both of these serum markers when we are trying to make a diagnosis of acute pancreatitis. Finally, in this module, I just want to point out to you that there are synthetic pancreatic enzyme replacements that patients can take as oral tablets. Children who are born with cystic fibrosis often develop pancreatic insufficiency. Their pancreas burns out and they can't make enzymes any longer. The same thing can happen in patients with chronic pancreatitis or patients who have had a pancreatectomy for any reason. There are multiple different commercial replacements available. They all have different ratios of lipase, protease, and amylase. But I just want to point out to you that when you meet people who have one of these conditions, you will often see that they are taking pancreatic enzyme replacement therapy. And that concludes our module on exocrine pancreas function.